Hi everyone, I'm İrem. I'm a bioengineer and currently I'm studying on my master's degree at the Marmara University Bioengineering program. Today I'll be presenting you the poster of our study. This study is a combined work of two departments, Professor Kazım Yalçın Arga, Dr. Ceyda Kula, and me. We are from the bioengineering department, and Associated Professor Mehmet Burak Aksu and PhD candidate Mehmet Mücahit Günce are from the medical microbiology department. So basically, what we aim here is to identify novel drug candidates against antimicrobial resistant Pseudomonas aeruginosa strains using a molecular profiling-based approach, and then to evaluate their effects on the clinical isolates. And today I'll be talking about our study topic, which is antimicrobial resistance, the experimental procedures that we used, and lastly, the results that we got so far. Antimicrobial resistance is the survival of pathogens when exposed to the antibiotics that once inhibited or eradicated them. It's a worldwide problem and also called silent pandemic in order to emphasize its importance. The overuse of antibiotics is one of the main reasons of antimicrobial resistance. Also, the pathogenic bacteria itself have resistance mechanisms as well, and biofilm formation is one of them. Basically, biofilm provides the bacteria to stay in a colonized form on surfaces, and this colonization protects the bacteria from external stresses such as antibiotics, and by that, the biofilm promotes the antimicrobial resistance. Bacteria that is used in this study, which is Pseudomonas aeruginosa, is known for its high biofilm capacity. It's a gram-negative and opportunistic type of bacteria, and it can cause severe infections when the host has a weak immune system. This is the main reason why it's one of the top risks for the patients in the healthcare services. So let's continue with the materials and methods. Medical faculty isolated the strains and applied several molecular identification methods to validate the strains being Pseudomonas aeruginosa. Later on, by disk diffusion method, they selected resistant strains and provided us a list of 10 resistant Pseudomonas aeruginosa strains for the study. We had to be sure that these strains have distinct genotypes, so we isolated the DNA of these strains, then made ERIC and rapid PCRs and run the samples on the agarose gel electrophoresis. In the similarity analysis, a paired group algorithm and Dice coefficient was used. Strains with an 80% or higher similarity percentage counted as similar, and one of them was eliminated from the study. After the necessary eliminations, we continued with the phenotypic assays to create the antimicrobial resistant profiles of these clinical strains. We applied MIC analysis, biofilm inhibition assays, and biofilm eradication assays. Since I don't have much time here, I'm not going to go over the details of the procedures, but you can always find the details from UCAST and take them as reference. For the RNA isolations, we used the MIC values that we found. We cultivated the clinical strains under sub-MIC and supra-MIC conditions, and by following the instructions of the RNA isolation kit, we obtained the extracts and later on started the RNA-seq-based transcriptomic profiling procedure. Our study still continues, so now I will be sharing you the results that we got so far. As I said, we first eliminated the similar strains. Here you can see our gel image and the dendrogram graph we made by using the base pair similarities. We eliminated 2 strains here, and later on we continued with the MIC analysis. You can see the MIC values here in this table as well. For amikacin, we couldn't find the specific MIC values for the clinical strain, so in the future we will repeat it again. After MIC analysis, we made biofilm assays as I mentioned, and here you can see the biofilm inhibition plate of imipenem at its last step. Stained wells stand for the biofilmed ones. Next to that, there is a biofilm inhibition profile of just one strain. You can see that at 0 concentration, when there is no antibiotic, there is a certain amount of biofilm. This shows that our clinical strain is able to form biofilm, but the maximum amount of biofilm formation is at the least concentration of the antibiotic, and then there's a dramatic decrease in the biofilm amount with the increase of the antibiotic concentration. This profile shows us the clinical strain reacted to antibiotic by forming more biofilm, but with the increase in the antibiotic amount, the formed biofilm was inhibited. With the antibiotics imipenem, levofloxacin, and piperacin tazobactam, we saw a successful inhibition against most of the strains. And in the case of biofilm eradication assays, none of the antibiotics were successful at eradicating an already formed biofilm. To conclude, I would like to emphasize on the increase of the resistance among the clinical strains. The reference MIC values for ATCC strain and the ones that we found for clinical strains have huge differences. Also, our strains are multi-drug resistant and their biofilms are not eradicated even with MIC times 4 antibiotic, but generally within supramic, biofilm was successfully inhibited, so we understand that the bacteria is not dead, but yet its biofilm is inhibited. So here we think that supramic doses not killed the bacteria but managed to downregulate the biofilm formation genes. And this is why we need the transcriptomic analysis to see how the genes are regulated and form an overall site to the study. I thank you all for listening and also thank the organizers of this conference for giving me this opportunity to present my work. I wish you all a good day.